So now I want to talk to you about the technologies we can use to measure gene expression and gene expression programs in single cells. So we'd really like to know what genes are being turned on in each of these T cell states. So we have this really amazing experimental technology where we can take a group of T cells and apply a technology called single cell RNA sequencing and read out for every single cell what genes are being expressed and to what levels they're being expressed. So the output of the single cell RNA sequencing experiment is this gene expression matrix, where we've measured the expression level of every single gene in every single cell. And so the rows of this matrix are the individual genes, and there are 20,000 genes encoded in the human genome. So there's 20,000 rows in this matrix. And then the columns are the individual single cells that we profiled. So there's 50,000 columns because there were 50,000 T cells measured in this experiment. Now, the actual values in the matrix are the expression levels of the individual genes inside a single cell. So here we can look at TCF7, and what this value means, 3, means that there are 3 RNAs that were counted from the gene TCF7 in cell 1, but only 0 RNA molecules counted from TCF7 in cell 3. And likewise, we can look at the gene GZMA, and we find that there are 0 counts of RNA molecules in cell 1, but 3 counts of RNA molecules from GZMA in cell 3. So once we have this gene expression matrix, we can actually start clustering cells together based on their similarities in what genes they're expressing. So for instance, we can see cells 1 and 2 are similar to one another because they both express TCF7 and BOC2 at high levels, and they have lower levels of all these other genes. Likewise, cells 3 and 4 express PRF1 and enzyme A, GZMA at high levels, but they have lower levels of all these other genes. And finally, cells 5 and 6 have high expression levels of PDCD1 and EOMES, but they have lower levels of expression of all these other genes here. And by clustering cells by their similarities in the genes that they're expressing, we can actually identify the programs and the cell states. So if you remember, TCF7 and BOC2, they mark the progenitor T cell state, and PRF1 and GZMA mark the effector T cell state. And finally, PDCD1 and EOMES mark the terminal exhausted T cell state. So from this data, we can really learn what the gene expression programs are. And remember, we're not just measuring 2 genes at a time, we're measuring all 20,000 genes that could potentially be expressed in a single cell. So that's kind of the output of a single cell RNA sequencing experiment, and we'll discuss how this works momentarily. But then the other technology we'll discuss is how do we measure the effects of genetic perturbations in single cells. So why do we want to do this? So ultimately, we want to take T cells and modify them in some way, make a perturbation in some way to make them better at killing tumors, killing cancer cells. Because right now, T cells in cancer normally enter this exhausted state, this terminal exhausted state, and are unable to kill the cancer. So we really want technologies to measure the effects of perturbations in T cells. So here's the T cell differentiation pathway we've seen before. And in this example, we're just going to change one gene, this gene called TOX, and we're going to delete it from the T cell. And in general, TOX deletion strengthens the anti-tumor T cell response. So deleting this TOX gene favors the progenitor T cell states and the effector T cell states over the terminal exhausted T cell state. So we're going to introduce this perturbation into the T cells, let the cancers grow up in the context of a tumor, and isolate the T cells, and then do this technology called Perturb-seq. And we're going to define Perturb-seq in the later slides. So the output of the Perturb-seq experiment is again a gene expression matrix where we have all the genes that are measured across the single cells, but we're also able to detect the presence of the TOX deletion inside the single cells. And the TOX deletion acts by affecting the expression of many other genes, and that is going to shift the distribution of the T cell states to favor the progenitor state and the effector state. And so now if we look at the gene expression matrix, we still see the genes that mark progenitor T cells and effector T cells, but the genes that mark the terminal exhausted T cell states, such as PDCD1 and EOMES, their expression level is close to 0. And that's because, again, this TOX gene is affecting many genes, and it's affecting the distribution of the T cell states. And so we no longer really are observing the terminal exhausted T cell state after making the TOX genetic deletion. These technologies have really taken off in the last decade or so. So what we're looking at here are different single-cell RNA sequencing profiling technologies, And these are on the x-axis are the dates in which the studies were published and the technologies were first demonstrated. And on the y-axis are the number of cells that were profiled in that by that technology. And what you'll notice actually is that the y-axis is on the log scale. So in 2009, the first single-cell RNA sequencing experiment was performed. This was on a mouse blastomere, which is a 4-cell stage embryo. And that was the first experiment done, single cells. And then now, or 5 years ago, in 2017, we reached the level where in a single experiment, we could profile 1 million single cells. So what this means is for 1 million single cells, we could quantify the expression level of all the 20,000 genes in those single cells. So this is really amazing growth, and it's really led to a revolution in many fields in biology. And it's also impacting our studies of cancer immunology and cancer immunotherapy. So now let's talk about how the single-cell RNA sequencing experiment really works. So we always start with a piece of tissue. So in this case, it could be a tumor like melanoma, and that tissue gets taken out of the patient, and then the tissue is made up of individual cells that are kind of working together and communicating in the context of that tissue environment. We take that tissue and we dissociate it into the individual single cells. And now, once the single cells are isolated, we want to extract their RNA, and the RNA is what tells us which genes are being expressed in the cell. And so by looking at what RNA is inside the single cell, we can identify gene expression programs and the cell states of these T cells that we extracted from the solid tissue. So I next want to give you an overview of how we isolate the single cells in the single-cell RNA sequencing experiment and label the transcripts. Now, this is really amazing and really cool technology, but the exact details of how it works are not critical for your ability to perform in this data science challenge. So let's get going. So what happens are single cells are isolated in water droplets using microfluidic chambers. So this is a blow-up of a microfluidic chamber. The cells are flown into the chamber, and they get encapsulated in these droplets. And you'll notice within each droplet, there's also these colored beads. And these colored beads are going to help label the transcripts from the single cell, and it will tell us which cell that transcript came from. So remember, transcripts are just the RNA that's being transcribed from the gene. And now all these RNA molecules in these single cells get labeled with a different barcode, which is really like a different DNA sequence. And here we have cells labeled with a purple barcode, a blue barcode, and a red barcode. And now when we read out those transcripts using sequencing in the next slide, we'll know which cell each transcript came from. And so now let's go back to this earlier cartoon. I just explained to you how single cells are isolated using microfluidics, and we're able to label the RNA. And then we take this RNA and we actually reverse the process of transcription. So if you remember from the introductory materials, DNA is transcribed or copied into RNA. We can actually reverse that process and make a complementary piece of DNA. And we do this because DNA is much more stable, and we can also make many copies of DNA, which is useful because then what we're going to do is sequence all those DNA molecules that were derived from the RNA. And sequencing is literally just a process of reading out the A, T, C's, and G's that are going to define that DNA. And once we have those sequences, we can actually identify what gene that sequence came from, because the entire human genome was sequenced, and we know the sequence of all individual genes. So when we have a DNA sequence, we can look up what gene does it match to in the human genome, and that will tell us that, okay, TCF7 was found in this cell, and PDCD1 was found in this other cell. And that's literally just by sequencing out the DNA that comes from this experiment and matching it against the human genome. And so the final outcome then are these big gene expression matrices, and let's look at them in a little more detail. So to remind you again, the rows of this gene expression matrix are the genes, and there's 20,000 genes we could measure. And the columns are the individual cells. And here in this experiment, we've measured 70,000 cells. And then the values are how much each gene was expressed in every single cell. So TCF7 expressed at level 3 in cell 1, GZMA expressed at level 0 in cell 1, but level 3 in cell 3, and so forth. So what you notice as well in this matrix is that there are many zeros, actually. And this is not a surprise because there are many biological reasons why a gene would be at level 0 in a cell. So Remember, all our cells are defined by gene expression programs. And these gene expression programs don't include all 20,000 genes. They include tens to hundreds of genes. These gene expression programs define what the cell is doing, and all the other genes are really not needed for what the cell needs to do in the tissue context, and so they don't get expressed on purpose. So the level 0 here is for a biological reason. However, the zeros also can arise from technical reasons because it's very hard to actually capture all the RNA that comes from a single cell. So you can imagine a single cell is very, very small. It's on the order of 10 microns or so. And the RNAs, there's many of them, and being able to capture all of them is difficult. So sometimes you just fail to capture the RNA and you get a 0 in that matrix. So once we have this gene expression matrix, now we start to do computational analysis to identify the cell states that we're finding in this experiment. And there are several steps in the computational analysis, starting from quality control and normalization of the data, and then feature selection, dimensionality reduction, and clustering. And so we're going to walk through each of these steps so you understand how we go from the gene expression matrix to actually identifying the cell states that are there, the gene expression programs, and learning what we actually see in the sample that we're profiling. So the first step in our single-cell RNA sequencing analysis is quality control. And the idea here is we want to remove low-quality data that we don't want to work with. And here I've ordered all the single cells by the number of genes that were detected in that cell. And the maximum for this axis is 20,000 because that's the number of genes in the human genome. So the cells are ordered by the number of detected genes. And you'll see there are two tails to the distribution. And we focus on those two tails. So this tail here, where a low number of genes are being detected, is often found due to low quality cells that have been damaged in the sequencing process. And so we remove those from analysis. And then this other tail here is often found because the single cells are actually still stuck to one another when they go into the droplet. And so it's not really a single cell we're looking at, but multiple cells. And so we also remove those from the downstream analysis. After that, after we perform quality control, we then normalize the data. So why do we normalize the data? So if we look at the total gene expression in a cell, we find that that value often varies for technical reasons. And the total gene expression in a cell is just taking the cell and looking at all the genes that are expressed in that cell and summing up all those values to get the total level of gene expression in that cell. And we find that that total varies quite a lot across cells. And this is often for technical reasons. And if we don't correct for this technical effect, this is a confounding factor in a lot of our downstream analysis. So what we end up doing is for every cell, we normalize the total gene expression. So now the total gene expression is just going to sum to 1. And then what we do is we multiply all of that by 10,000. So now this total expression sums to 10,000. We add 1 and take the logarithm. And this is a standard kind of approach that's done in a lot of these sequencing studies. And the reason we're going to be adding 1 is, if you remember, a lot of these gene expression values are 0. So if you take the logarithm of 0, you know that's bad. So we add 1 to avoid doing that. So now we've done the normalization, we move on to the feature selection step. So I first want to start with this cartoon of cells in this sort of three-dimensional space, where the axes are the expression levels, how much each gene is found inside those cells. So we have gene 1, gene 2, and gene 3. And there's a group of cells here, here, and here. And they're kind of all in these different kind of clusters where these cells have high gene 1 and 2, these have high gene 1 and 3, and these have high gene 2 and 3. So this is an understandable way to look at cells and their gene expression. However, unfortunately, or fortunately, depending on how you look at it, cells actually exist in 20,000 dimensional space, where they don't just express 3 genes, they could express up to 20,000 genes. So this would be very hard to imagine, like humans cannot visualize higher dimensional spaces. However, this is really the space in which the cells are existing. And so this is hard to visualize. It's also hard to work with from a machine learning perspective. It's hard to cluster things that are in very high dimensional space. So what we do is we do feature selection, where we only focus on those genes or features that have highly variable expression across the cells. And the idea here is that genes that are variable in expression are genes that are going to explain the cell states that we're seeing. So let's think about a gene whose expression level is the same across all the cells we're seeing. So that's not going to help us differentiate a progenitor T cell from a terminally exhausted T cell because the gene's expression level is the same across those states. However, if a gene is variable, meaning its expression is high in one cell state and low in other cell states, that's actually informative about what differentiates the cell states. So for instance, you remember the TCF gene is highly expressed in the progenitor T cells, but lowly expressed in the terminally exhausted T cells. So that's an example of a gene with variable expression across the cells. And so what we do is we take these 20,000 genes and we select the 2,000 to 5,000 most highly variable genes. And that's our feature selection step. And so at this point, we've gone from describing our data with 20,000 genes or features to 2,000 to 5,000. So this is a reduction in the dimensionality of the data. And now we can go even further with that. So now let's pretend, or we can imagine that our cells are in 5,000 dimensional space after this feature selection, and we want to go even lower. And we're actually powered to do this because let's return to the idea of our gene expression programs. So remember, in the gene expression programs, you have groups of genes that are turned on or off together. So if I have a gene expression program that defines, say, the progenitor T cells, if I tell you that, TCF7 is high in these T cells, that actually tells me a lot about all the other genes that are in that gene expression program because they're turned on together. So knowing the expression level of one gene actually tells me a lot about many other genes that are being expressed in that cell. And so because of that, genes are really not independent of one another. Measuring one gene can tell you about many other genes. And this is really the key that allows us to go to even lower dimensionality spaces when describing our data. And so a really common technique is this linear dimensionality reduction technique, principal component analysis, or PCA. And this allows us to move from describing cells with 5,000 genes to this new space where cells are described with 10 to 100 principal component scores, where these principal components are really reflecting the different gene expression programs within the cells. So at this point now, we've gone from 5,000 dimensional space to about 10 to 100 dimensions. And this is a lot easier to work with when it comes to clustering to identify the T cell states. So the last step in the analysis is we've done this dimensionality reduction using principal components analysis, and now we're going to do graph-based clustering. So we're in the principal component space, and we're going to calculate the distance between every pairs of cells. This is the Euclidean distance between cell 1 and 2, cell 1 and 3, and so forth for every possible pairs of cells. And then we're going to use that distance matrix to build a k-nearest neighbors graph. So here are the individual cells in sort of this lower dimensional space that we've moved into. And we're going to ask for every cell, who are my k-nearest neighbors? And we're going to build that graph. So for instance, this cell, the nearest neighbors are here, here, and here. And we do that for every single cell, and we build that graph. And now, once we have that graph, we start to cluster in that graph space. And you can already by eye, where would you draw the clusters? You would probably draw a cluster here because there's a lot of connections between these cells, a cluster here, and a cluster here. And there are many clustering approaches out there. One of the most common clustering algorithms is the Leiden algorithm. And this is an algorithm to detect communities within graphs. And this algorithm works by optimizing a metric called modularity. And so we do Leiden clustering, and we identify 3 clusters in this toy example. OK, so at this point, I've gone over with you all the steps in a typical single-cell RNA sequencing analysis. We started with quality control and normalization, and then we went to feature selection, dimensionality reduction, and this graph-based clustering. And now let's apply everything we've learned to the actual data that's in the data science challenge. And so this is data collected by the physician-scientist Mark Schwartz and Neerha Cohen at the Broad and Dana-Farber Cancer Institute. And in the challenge, we have T cells that have been collected from mice with melanoma. And we're showing all the T cells in this plot here. And this is what's called a UMAP embedding. And the UMAP embedding is a visualization approach to take high-dimensional data, which is existing in this high-dimensional space, and showing it in a nice 2-dimensional plot. And so the UMAP embedding is calculated from those principal component scores that we introduced in the previous slides. And the idea with the UMAP embedding is it's trying to preserve local distance similarities. So if 2 cells are similar to one another in the high dimensional space. And by similar to one another, I mean that they have similar genes being expressed. So if they're similar in the high dimensional space, then we want them to be close to one another when we visualize them in 2 dimensions. So what this means is like these 2 blue dots here that are really close to one another. In the high dimensional space, they were close to one another. And so when we plot them in 2 dimensions, we want them to also be close to one another. So this is a separate embedding that gets calculated, and you can read more about it here, and it's calculated from the principal components. And then we take that embedding of all the T cells, and now we color the T cells by the identity of the clusters that we learned in the previous slide. So remember, in the previous slide, we identified about 3 clusters. Here we have 8 clusters, and they're all colored differently. And what we see is that there's this really strong clustering in the data where these T cells look like they're one cluster and these T cells look like they're another cluster. And this is really what's going to lead us to identifying what the T cells are doing. So the idea is that T cells in this cluster are having one set of gene expression programs that's distinct from T cells in this cluster over here. And so to get at that, we start to look at what genes are being expressed in each of these cells. So remember, every single cell here, in theory, we have up to 20,000 genes measured. So we can take cells in one cluster and ask what genes are more highly expressed in this cluster relative to all the other clusters. So for instance, if we take the progenitor T cells in this kind of brownish orange color, we can ask what genes are more highly expressed. And so what we would see, or what we do see in the actual data, is the gene TCF7 is higher in this cluster relative to all the other clusters. And that clues us in that this T cell cluster is actually reflecting the progenitor T cell state, and we can label it progenitor. Likewise, if we go over to this cluster of T cells over here, and we look at what genes are more highly expressed, meaning they're at higher levels than all the other clusters, we see the gene PDCD1 coming up. And remember, this is a gene that marks the terminal exhausted T cell state. So by doing this game for every single cluster, we can actually annotate every cluster and say what T cell state these cells belong to. And what's really beautiful is that we see all the T cell states that we would expect to see in the context of a cancer, in this case, melanoma. So we see those progenitor T cells, we see the effector T cells, we see the terminal exhausted T cells, and we see the cycling T cells. So at this point, now you have an understanding of how single-cell RNA sequencing works and how we can take a population of cells, measure the gene expression program within the single cells, and with some analysis, identify the gene expression programs and the linked cell states for those single cells. So the next problem that we really want to solve is what happens when we do genetic perturbations to the T cells, and how does that affect the gene expression programs? And again, the reason we care about this is because currently, regular T cells enter this exhausted state and are unable to kill the tumor. So we really need to make some change to the T cell to enable it to better kill the tumor, to kill cancer cells. So what we would like are technologies that allow us to test perturbations in cells and see what happens to the gene expression of those cells and the resulting cell states.